DMSO has been the gold standard because of its unique chemical properties and extensive clinical track record, making it difficult to fully replace. But that's a 60 years old product, and a lot of things have changed since then. So some of the properties are as follows: physical chemical efficiency. It has a rapid membrane permeability, and it enters the cells and equilibrates with the intracellular environment faster than other penetrating agents like glycerol. Water displacement, it forms hydrogen bonds with water about 30% stronger than between water molecules themselves, thereby preventing the formation of intracellular ice crystals. It's versatile in the pre-cell therapy days.
For 60 years, one molecule has defined cell cryopreservation, effective enough to become the universal standard, yet problematic enough that the FDA attempted to ban it twice. My guest today, Steve O, spent 22 years at Singapore's A-Star, invented stem cell microcarrier technologies, CRISPR activation, and a lot more. He holds 43 patents and has seen this problem from every angle. Steve brings hard-won wisdom to one of bioprocessing's most persistent challenges. Why has DMSO survived this long? And what finally threatens its reign? Let's find out. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brohlman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, Steve. It's good to have you on today.
Thank you, David, for the invitation.
Steve, shares something that you believe about bioprocess development that has made the most impact.
Okay, I'd love to. I think there are three that I can think of right now. The development of the AMBER system for a small-scale volume optimization that has made a lot of impact in the biologics industry. The development of perfusion high-intensity cultures that has reduced the volume and the footprint of bioreactors for generating high titers of proteins, recombinant antibodies. And I think one that's coming in the future is the use of digital twins to reduce the numbers of experiments and then focus on the key experiments. I believe you're going to be talking about that in the future, right?
Yes, absolutely. And we have already covered a few times this topic, digital twins, hybrid modeling, on a few episodes. So if you're interested in that, Smart Biotech Scientists, go back and listen to these episodes. It's a huge pleasure to have you back on, Steve. For those of the listeners who had not had a chance to listen, our interview we did about, I think it was a couple of years back. Take us back to the beginning. What sparked your passion for biotech and cell therapy, and what pivotal moments during your long career shaped your path to becoming a leader in stem cell bioprocessing solutions?
I think back in the 90s, when the production of antibodies was challenged, I had the opportunity to complete my PhD and get into learning about use of bioreactors for manufacturing cells, which at that time was a big challenge. People were making bacteria fermentation, but not animal cells. So that was the first pivotal moment to give me the training and the opportunity to get into this field. And then around 2001, post the discovery of pluripotent embryonic stem cells, again, that was a challenge to now how to produce stem cells at scale in the bioreactors. And we built a small team to look at growing these cells, which were very dependent on support cells, feeder cells, matrigel, all kind of undefined conditions. And finally, we found a way to switch them to grow on microcarriers and bioreactors. So those were the two kind of big opportunities that I had to make an impact in bioprocessing and come up with new solutions to scale.
And what were some pivotal moments you experienced your long years at A-Star that finally led you to also take a leap and pursue entrepreneurial endeavors?
After the discovery of microcarriers for manufacturing stem cells, I did try my hand in forming a spin-off company, but the challenge was raising cash to do a CDMO type of business model and then finding a CEO. So my entrepreneurial skills was not that strong and we didn't succeed. Glad to see that there are companies out there now that are able to make that impact. Still no products made out of bioreactors yet, but people are trying.
Wow, that's fantastic. You hold a lot of patents. It's exactly, if I'm not mistaken, 43 patents across microcarriers technologies, serum-free media, and CRISPR activation. So you could have focused on many different areas. What drew you to cryopreservation as one of your key advisory roles? And tell us also what made you believe that this was an area ripe for innovation.
So it was in collaboration with a company called XTherma back in 2015, I believe, or maybe a bit later than that, where I saw that they had this amazing solution that could transport whole hearts across the country over 3 days. And we had seen there were challenges with DMSO for cryopreservation of pluripotent stem cells and the first challenging differentiated cell type called neural stem cells. So I approached the CEO, Xiaoxi, and looked at potentially testing out that solution for stem cell applications. And I realized that in the final cell therapy, you wouldn't be giving fresh cells. You'd have to freeze them down and use the thawed cells in patients' injection. So we needed something that would be robust and give potent cell properties as close to the fresh cells as possible. And really nobody at that time was looking at the replacement of DMSO, the standard solution. So that's why I felt that there was a blue ocean opportunity to collaborate with them. And then once I left A-Star about 4 years ago, I sought the opportunity to be a scientific advisor. That's why I'm working with XTherma on educating the field in alternatives to DMSO.
Let's start with the fundamentals, Steve, because cryopreservation, or shall I say the preservation of cells, is a big, big topic, not only in cell therapy, but I'd say in various areas of biologics. Help our listeners understand what are the main challenges scientists face when preserving cells, freezing cells, and why has DMSO remained the gold standard despite its limitation for many decades?
So DMSO has been the gold standard because of its unique chemical properties and extensive clinical track record, making it difficult to fully replace. But that's 60 years old product and a lot of things have changed since then. So some of the properties are as follows: physical chemical efficiency. It has a rapid membrane permeability and it enters the cells and equilibrates with the intracellular environment faster than other penetrating agents like glycerol. Water displacement, it forms hydrogen bonds with water that are about 30% stronger than between water molecules themselves, thereby preventing the formation of intracellular ice crystals. It's versatile. In the pre-cell therapy days, hematopoietic stem cells were mostly the cell type that was used in cryopreservation blood products, some immune cells and MSCs. And the HSCs have been used in clinical and regulatory environments, so lots of stem cell transplants. Its side effects are documented and manageable within existing medical protocols, such as a maximum daily dose limits, typically 1 gram per kilogram of patient weight. There are standardized protocols, workflows built around the 5 to 10% DMSO formulation. Transitioning to alternatives requires a lot of extensive and costly validation so that the therapeutic efficacies are not affected. And then the practical and economic advantages includes low cost, high stability, shelf life, and temperatures. They're widely available in high purity grades, US pharmacopoeia for clinical use. And it's easy to use in the control rate freezers at -1 degrees, so fairly simple to implement. So that's the reason why they've been in use for 60 years.
What I'm hearing here, Steve, is that DMSO has a lot of advantages, and that's the reason why it has been used for so many times. Isn't it interesting that the FDA has tried to ban DMSO twice? So what are the specific issues with this toxicity of DMSO, and what are some other aspects that make the use of DMSO problematic?
So the example was back in 1965, there was a ban on human testing because of safety alarm regarding lens eye toxicity in animals, and then the sudden death of an Irish woman after topical use. So these early concerns established regulatory wariness that persists today. Now, for the modern cell therapies that are new issues, we've never had the challenges of the variety of cell therapies that are now around. This include cellular dysfunction and loss of potency for T cells. At concentrations as low as 0.25 to 1%, DMSO inhibits CD4 positive T cell activation, proliferation, and cytokine production like IL-21, IL-4, and IL-17A. It downregulates genes in early signaling and T cell receptor pathway. Then for NK cells, DMSO is associated with altered expression of the natural killer cell markers and diminished effector functions. For stem cells, DMSO is known as a differentiation inducer, so it can downregulate pluripotent factors like OCT4 and NANOC at concentrations as low as 0.1 to 5%, potentially biasing their therapeutic identity before they reach the patient. In terms of physical and structural damage, DMSO induces pore formation in cell membranes and can disrupt the cytoskeleton by dehydrating lipids and interacting with proteins. In terms of mitochondrial and metabolic stress, it can compromise mitochondrial respiration, induce oxidative stress, and trigger apoptosis through caspase 9 and 3 activation. And then in patients, DMSO toxicity significantly increases dose-dependent cell damage and adverse patient reactions. Clinical risks include cardiovascular issues, severe nausea, neurological symptoms, allergic reactions, which necessitate swift removal or lower concentrations.
Let me reframe this because you're making an excellent point here, Steve. I have worked in biologics for most of my career, and DMSO is used to freeze cell banks. And pretty much the only parameter many other teams were interested in is the viability, so when you thaw your cells. But now what I'm hearing is that in cell therapy, we are playing a completely different ballgame because we're not just cultivating cells in a bioreactor. Now we are using these cells to treat patients. So we have to look much more carefully what's going on at the genetic level, at the transcriptomics level, at metabolomic level. So we have a plethora of aspects to watch out for.
Correct. Because the cells are the functioning entities. Just having viable cells is sufficient. You have to have the cells be able to do the function that they were made for, as when they were fresh.
Can we look into the cells a bit, what's happening when you're using DMSO and you're freezing and then thawing the cells? What exactly happens, I'd say, really at the microscopic level? What are these changes that affect the genetic stability or the transcriptomics or the metabolism of these cells?
Some of the toxicity issues in cryopreservation is that when the cells are exposed to DMSO at temperatures above 4 degrees during thawing, it can disrupt the cellular membrane, cause mitochondrial damage, and lead to the production of reactive oxygen species. So these kind of accumulative, and they can start to create all those metabolic damages that I mentioned earlier. On top of that, exposure to this DMSO can lead to undesirable phenotypic changes in the stem cells due to the alteration of the DNA methylation and histone enzymes that can open up the DNA for priming towards differentiation or close it down such that they can't differentiate. Then time and dose dependent risk. Toxicity is directly related to concentration and exposure time. So typically they're used in the 5 to 10% range, but even up to 40% DMSO has been used, and this is completely destroy hemaprogenitor cell viability. And then, as I mentioned earlier, the product, when residual DMSO is infused into patients, you have nausea, vomiting, cardiovascular events like bradycardia, arrhythmia, neurological symptoms like seizures and dizziness, and allergic reactions. And finally, in terms of the manipulating of the DMSO itself, because of these toxicity issues, they need to be washed away to remove the residuals and then reducing the overall volume to decrease the dose. This can lead to potential opportunities for contamination of the product by having to do this additional step.
To what extent can these detrimental effects happen even though the viability looks okay?
So even at low concentrations post-washer, the types of disruptions that can happen are as follows. Alterations in DNA methylation. DMSO disrupts the balance between the writers and the erasers of DNA methylation, leading to widespread genomic instability. On the one hand, hypermethylation. DMSO can upregulate methyltransferases like DNMT3A or 3B, leading to global hypermethylation in cell types such as fibroblasts or embryonic body. And this reduces the transcriptional activity by silencing the gene. Then the opposite thing can also happen. Hypomethylation can induce active demethylation by promoting the TET family enzyme TET1 and TET2 which convert 5-methylcytosine to 5-methylcytosine C. So the major risk in preserving stem cells and related products is we don't know how these methylation changes will have long-term effects on the cell product.
Given what we've discussed so far, Steve, what are the options we have at our disposal as a scientist? Because we could use DMSO, but there's some detrimental effects you just mentioned, and we have to be aware of that. And there are now some alternative products. So what are these products as scientists listening can use?
So there are a couple of approaches that have been taken. Companies have looked at trying to reformulate alternatives to DMSO by other small molecules like glycerol and other small preservatives. And then that's complex formulation that has been optimized with each cell type. And then there's companies like XTherma, which come up with an antifreeze peptide mimic, which is a single molecule that does the same thing as DMSO, but it has processing benefits and it doesn't have the significant toxicity that has been seen in DMSO. So those are the two approaches, either complex formulations or a completely new entity to replace DMSO.
And looking at XTherma, what I saw that you took inspiration from antifreeze proteins found in Arctic fish. How did this come about? Explain how these specifically protect the cells from freezing damage.
So these antifreeze proteins improve cryopreservation by binding to the ice crystals, inhibiting the growth of recrystallization and reducing damage to cells at sub-0 degrees temperature. They were derived from these cold-adapted organisms and create a thermal hysteresis gap that lowers the freezing point without affecting the melting point and protecting biological samples and enhancing survival rates. Essentially, how it works is that they will cluster around water molecules and prevent rapid growth of crystals. And because of these much smaller crystals, they don't damage the cellular structure. Either when they're frozen or when they're thawed, they would be able to become liquid again faster than large crystals without the antifreeze protein.
We've just unpacked why DMSO has persisted for 6 decades despite its well-documented limitations, and why that persistence has real consequences for your cells, your patients, and your manufacturing process. In part 2, Steve O takes us into the solutions, the science behind next-generation cryoprotectants, the data that's turning heads, and what a DMSO-free workflow actually looks like in practice. If this episode added value, please leave a review on Apple Podcasts or your preferred platform. This enables more scientists like you discover the show. Thank you for tuning in today. See you next time. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.